Have you ever wondered what you're missing when reading a research article? Although we may think we're getting everything we need to know about the research design, the methods, even the analysis, chances are there are key details missing from the paper. Details that are necessary for assessing the validity, reliability, and generalizability of the results. In today's episode, Mike and Joey speak with Dr. Arianna Gard about her work investigating reporting practices in neuroscience research. Dr. Gard is a professor of psychology at the University of Maryland, where she also participates in both the Neuroscience and Cognitive Science program and the Population Research Center. So, what's missing? Let's find out. Welcome back to another episode of Cracking the Shell on Psychological Science. I'm Mike Doherty.
And I'm Joey Barnett.
And today we are here with Dr. Arianna Gard. Welcome to the show, Arianna.
Hi, guys.
You published this paper recently in bioRxiv.
Sure.
I'm going to sum it up with just a one sentence, one-liner.
Great.
Well, it's going to be a question.
Okay.
So why don't you just tell us what's missing from neuroscience?
Yeah, lots missing. There's a lot of promise in neuroscience. So it's always good to start with a positive note, right? So neuroscience human neuroimaging in particular, has really shaped public ideas about who we are, has been involved in court cases like Simmons versus Roper, which refers to juveniles being competent to stand trial under age 18.
How much is this? I'm sorry, I'm kind of skipping ahead here.
But
So how much of the neuroscience is necessary for establishing that?
This is a good question, Mike. I think that for decades, scientists have been unable to satisfactorily convince policymakers of the importance of some of the issues that we care about.
So behavioral science is not convincing?
Behavioral science has not been routinely convincing.
But if I can show a picture of the brain, I can say...
there are studies that show that if you show images of MRI, of other neuroscience, that jury members, policymakers find it more convincing and more appealing. In addition, if America is going to be the STEM capital of the world, we're going to advance knowledge and science, then neuroscience is a key part of that. And so studying the brain is also about understanding who we are and about educating the next generation.
Yeah.
I mean, I will say that as I've gone on in my career, as a graduate student, I was like, neuroscience is going to change the world. But, I think it's a tool. I think it is a tool. I think it's not the end all and be all. I think that if anything, learning from community members that there are lots of methodologies that can bring a lot of rigor and depth into our understanding of humans.
Yeah.
Yeah.
Well, I'm just going to throw this out there, and then I'm going to leave this topic alone. My former student, Allison Roby, and I wrote a paper on the bridge between neuroscience and education, and we titled it A Bridge Astray. The question being, how much do we need to know about the brain in order to inform educational practice?
Sure.
And arguably everything that we know that's of practical use is behavioral.
Sure.
Right. And oftentimes, you can show changes in the brain or brain-behavior relationships, but if it doesn't impact the classroom, does it really matter? And I think that's sort of just worth pondering occasion. But we're not really here to talk about that today. I want to talk about your recent paper in bioRxiv.
Sure.
Because I found it quite fascinating. You raised a number of questions about, usually we talk about what's in the paper. But what you've done is you've sort of flipped that and said, what's missing from the paper? And so you've cataloged across, I don't know how many publications or how many manuscripts you looked at, but you've cataloged things that are there, but also things that are systemically missing. Do you want to say a little bit about that?
Yeah. So, basic research methods, we are taught to report on study sample characteristics, on how we recruit study samples, on how we drop participant samples, all in the name of reliability, reproducibility, and generalizability. So we want our data to be reliable to the true population parameter of interest. We want our data to be reproducible, so other scientists can reproduce what we try to study.
Absolutely.
And we want our data to be generalizable. That means it extends to a broader population of humans beyond just the sample that we study. And so in order to do that, we need a lot of methodological details. And so the paper is quite simple. It asks whether it was up to about 1,000 papers that were published in top-ranked journals that published neuroimaging studies. To what extent did these papers report all of these components of methods that are in our handbooks that we teach our undergraduate students, right? And so, what was so surprising was that basic, fundamental methodological details were missing.
Wow.
Including, for example, even something as simple as age. So you can report the average age of your sample. You can also report the range of your sample, right? And so most papers reported only one or the other. So are there 81-year-olds in the paper? Not sure, because the average was about 40 age or whatnot, right? So things like that.
Why would I care?
Sure.
I mean, the brain's the brain. I'm being a little
Is
facetious.
your brain as efficient as my brain, Mike?
No. I think anybody who knows me, that my brain has slowed significantly since.
Right? We can all just think back to our lives as we've aged, that the brain changes naturally with over time in lots of different areas, not just memory, but emotion. And think about a toddler, right? I have a toddler.
Same.
The ability for her to control her emotions is very different than my ability to, sometimes control my emotions, right? And so that's why we need to have just basic fundamentals of age, right?
Yeah, totally.
But we were also thinking about things like the sample's socioeconomic resources. So something about income or education or where people lived. Many studies have shown that the number of resources that you have shape how you experience the world. And when we think about neuroimaging, we're not talking about basic molecular processes like cellular division. We don't image. We can't take pictures of the brain at that level of inference. We're combining lots of cellular processes in big regions. And so, we can't, even though somebody with lower or higher educational attainment might have the same basic cellular processes, those large magna scale processes are going to differ between people.
Can I put you on the spot?
Yeah.
I don't know enough about neuroscience
Sure.
myself, but I'm curious. So you raised the question of the relationship between, say, socioeconomic status and brain development,
Sure.
brain functioning.
Sure.
Give me an example of that relationship. Like,
Sure.
what changes between, say, high and low socioeconomic status in the brain?
Sure. So maybe one of the best examples is the link between income, childhood income, and inhibitory control. So our ability to resist a automatic response.
Okay, that's the behavior.
Sure, that's
Okay.
the behavior.
I want to know the brain.
Yes, the brain. So those same regions that are responsible for inhibitory control receive less activation in those regions while somebody is doing one of those inhibitory tasks for folks who come from environments of lower or higher income.
Okay, what is that? Dorsolateral prefrontal cortex?
Inferior frontal gyrus.
Okay, we'll just edit out my stupid.
And the degree of activation in the inferior frontal gyrus relates to behavioral performance on inhibitory control tasks, right? So the more, in this case, using fMRI, oxygenated blood that reaches the inferior frontal gyrus, the better you are able to resist those automatic prepotent responses.
What's the mechanism for that?
Well, that is one question with fMRI, that we are measuring changes in oxygenated and deoxygenated blood. And so changes in blood flow is presumed to reflect neuronal activity. But in human fMRI and human MRI, we do not measure single neurons, right? And so there is a level of inference that we're making when using these methodologies developed for humans and trying to say something about neural function.
Yeah. These things are complicated for sure. People talk about psychology being a soft science. It bothers the heck out of me.
God.
It is hard.
It's
and
very hard,
you just think about the dimensionality of all the neurons in the brain.
And also like the complexity of income, right?
Right.
Low income means a lot of different things.
Oh, it absolutely does.
Yeah.
All right, but so far you've only touched on a few things.
Sure.
Some of the methodologies.
Sure.
What else is missing?
From these papers.
From these papers, yeah. What did you find?
Yeah, so in addition, there were very vague statements of why some participants were included or excluded in studies.
Ooh, that's bad.
Which is bad, right? So in an MRI, this was a study of MRI and fMRI papers. The machinery that's driving our ability to measure deoxygenated and oxygenated blood is a giant magnet, right? So in part, people can't go into the MRI if they have any metal in their body, for sure. But there are other reasons why people may not want to go into the MRI. They might be scared. They might be nervous. They might just flatly refuse and not really tell you why.
Any worry that these might be like collinear with like income or education?
That's exactly right.
Okay.
Right. And so there's some collinearity between the reasons why people don't participate in MRI and the very variables that folks are studying.
So this seems problematic from a generalizability perspective.
Exactly.
Okay.
And so at the minimum, we would even want to know what some of these reasons why somebody didn't get into the MRI. So for example, early on in early imaging studies, we found that much of the data that is not usable in the final analyses is because participants move a lot, right? So MRI is like taking a old school picture where if you move, the image blurs, right? And so in studies of, for example, children are not very good at staying still, right? So children are quite difficult to image. And what this recognition produced is lots of technology that has been focused on training kids for their MRI scans. So if we hadn't have been recording that information of why kids' data were being thrown out, we wouldn't have had this more newer technological advancement that has improved the retention of data. And so the same can be said about other reasons for non-participation in MRI research. And so, there were about 50% of the papers didn't explicitly say participants were excluded or included if blah, 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 blah.
Yeah.
Well, that's really, really interesting. I mean, this is, we teach, undergraduate students the importance of including that. So that's pretty surprising.
And, you saw statements like participants were left-handed and free of health challenges. What's a health challenge?
Like,
what does that mean?
For being excluded? Or
there was no word excluded. Participants were left-handed and free of health challenges.
That's interesting.
Right? No eligibility, excluded, included, none of that. Right? So from not only from a generalizability, but from a reproducibility, right? How am I supposed to recruit participants consistent with your study if I don't even know why you included or excluded people?
Are you familiar with this recent not meta-analysis, but big study where they tried to replicate a bunch of cancer studies? I think it was done by OSF or the community of, what is it called?
Open Science Foundation.
Open Science Foundation, yeah. So led by Tim Arrington and the crew.
Yeah.
They found very similar effect. I mean, the reporting of details of studies was
very bad.
Very bad.
Yeah.
So bad that they couldn't actually engage in replication.
Right.
Like they wanted to try to replicate all these studies. And, on top of the fact that they could only replicate, I think it was about 40 to 48% of those studies, like the findings could be replicated.
Yeah.
About half of all the studies they intended to replicate, they couldn't even do because the details weren't there.
Right.
And then even when they reached out to some of the authors, they couldn't get the details, whether they could have been lost or.
Right.
So, I mean, did you Like, how does this map on? I mean, it seems like very similar finding to what you're finding.
Very similar finding, just specific to neuroimaging. And I think what's unique about neuroimaging is that neuroimaging data is quite complex. You can imagine there are millions of neurons in the brain, and we have lots of data points. So neuroimaging data is kind of four-dimensional. So if you think about the brain as divided into a number of cubes, hundreds of thousands of cubes, and then you have time. So while people are doing something in the MRI, we're taking a snapshot of all of those 100,000s of cubes. And so reshaping that data into something that's usable involves a lot of steps. It's not that this isn't the case in other types of data, but neuroimaging data in particular really suffers from something called researcher flexibility, analytical flexibility. Okay. And so the more different options, should you clean it this way or clean it this way, every single decision that you make, something that I know, Mike, you know about, is the garden of forking paths, right? There's endless options for cleaning said data. And so in terms of the reproducibility of this work, documenting these methodological decisions is very important. But what my paper talks about is, yes, that is important for reproducibility, but we also have to know who's in our samples and how we're recruiting patients, right? So a brain is not a brain is not a brain is not a brain, right? Every brain is different and unique and special, and that's what makes humans wonderful, but also complex. And so, that was one big conclusion of the paper. And the other conclusion of the paper is that there's been a very recent and appropriate push for very large sample sizes. So typically, as you mentioned earlier, effects of the environment on the brain or brain on behavior are typically quite small, right? So meaning that there are lots of regions that contribute to something like anxiety, right? Not one region of the brain. So we need to look at that oxygenated blood flow across the entire brain and how regions interact with one another. And so in order to detect those small effects, we need lots of people. And so this push towards large sample sizes, you might think, well, the bigger the better. I have been in discussions with big neuroscientists who say, well, we just need bigger samples. That's the answer to generalizability, is we just need more and more people. But you can have a sample of a very, for example, middle class from College Park, right? 1,000 people that won't generalize to even across the state line to DC, right? And so bigger is not necessarily better. And what we found in this is that bigger does not necessarily translate to more transparency. So bigger studies were not reporting more information about their methods. In fact, it was a precise zero. There was zero association between sample size and transparency. On the other hand, the larger the sample size, the massive increases in how often papers were cited by other scientists, whereas transparency, the likelihood of somebody reporting these methodological features, was also basically associated with zero relationship with citations by others. So it's really an idea of how much is this basic practice of scientific methods valued by the scientific community, and I think that this also ties back into the other work in the Gard lab, which is that if we want people to take up science as important for them, science has to capture their experiences. And we may not be doing that if we're recruiting a very small slice of the population, or even worse, not even reporting who's in the sample.
Yeah.
So, some of my future work, I am really interested in when community members hear about scientific studies in news articles or whatnot. If there's information about how representative the sample is, do they believe it more? Or does it not matter? My hypothesis would be that folks do believe it more because it's maybe more rigorous.
Yeah, very interesting.
Yeah.
Well, arguably, a case is to be made for the necessity of research transparency.
Sure.
And, documenting everything that we do. I mean, this is like a foundational assumption of the work we do, right? Is that we are rigorous, we are transparent. And it's unfortunate that so much stuff, and it's not just neuroscience, but there's so much stuff that's published that just does not include the necessary details. I have to believe that some of this is due to the journals and other just kind of like we have not been good about what we report and how we report the work. And clearly the reviewers aren't doing their job when they're not picking this stuff
up.
Well,
they're, reviewers aren't getting paid either.
I know.
So
That's
all a system, Mike.
Don't get me going. That's going to turn this episode into a 45-minute soapbox presentation. Arianna, very good to see you again.
Thank you for having me.
Thank you so much for coming on.
Sure, anytime.
Yep, we might have you back.
Okay.
All right, cheers. We are live again here on Cracking the Show. Let's see. Welcome back to Cracking the Show. Let me try that again. Welcome back.
Third time's a charm.
Welcome back to another episode of Cracking the Show on Psychological